So hello, everybody. Welcome. Welcome, Jake. Thank you for 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 fighting with your internet and getting here. We're really excited today to have Jake Estes with us. He's a DARE investigator. He's one of my favorites. His work is always so wonderful and so interesting. So you know that we're doing this series on the HIV reservoir, which is very complicated. And, we all thought that we really needed Jake here to explain the reservoirs and the differences and all the work that he's done to find out so much about different sites. So Michael will be handling the chat, and please put your questions in the chat, put your name in the chat so we can welcome you. And Michael will pick up at the end, and then we'll have a question and answer period once Jake is finished with his presentation. And then after the general questions, Marc Wagner and I will ask Jake some questions about the presentation as well. In the middle of maybe 3/4 of the way through, Michael will send an interactive question so you can tell us what you thought of the webinar, whether it would met your expectations or whatever. And then for the next webinar, that will be, oh, I'm sorry, I usually have this right out. I'll announce it at the end, okay? So it won't waste anybody's time trying to figure it out. So Jake, take it away.
Thank you. It's always such a delight to present at this webinar. It's one of my favorites, and I appreciate the invitation. So what I'm hoping to do today is talk to you about where the HIV reservoir resides. And I want to emphasize the plurality of this reservoir, and I'm going to talk about the plurality of this reservoir today. Let's see. There we go. So I want to start with kind of my community summary. The major question that my lab really focuses on is, where do HIV reservoirs reside throughout the body from which infection could rebound after ART is stopped? What I hope to talk to you and convince you today is that before and during antiretroviral therapy, using an SIV model for HIV infection, that reservoirs are distributed throughout the body, but predominate in lymphoid tissues, particularly the gastrointestinal associated lymphoid tissues and the lymph nodes and other secondary lymphoid structures. There are multiple forms of reservoirs. Obviously, we hear a lot about latently infected reservoirs, but there are active reservoirs, those that are transcriptionally active. And while these active reservoirs decrease substantially in most tissues during antiretroviral therapy, there are some tissues in which antiretroviral therapy does not seem to impact the quantity of these particular cells. Active viral reservoirs remain detectable in all tissue compartments during antiretroviral therapy, while at a reduced level in most tissues, they are found throughout the body. But the dominant source of these active reservoirs appears to be the gut. And then I'm going to talk a little bit about a unique and understudied uninfected cellular reservoir called a follicular dendritic cell network. And I'll talk about the importance of this uninfected cell reservoir in the context of storing and for long periods of time, potentially infectious viral particles. Why is this important? These data highlight the very large size and distribution of infected and uninfected HIV reservoirs throughout the body. And the large size, diverse distribution, and distinct nature of HIV reservoirs within tissues throughout the body underscore challenges in developing effective HIV cure strategies that must target multiple sources of potentially infectious virus. Why should we be excited or not about this? This is kind of a yin and yang. I mean, the data I'm going to talk to you are pretty daunting. Point to some daunting challenges because of the size of HIV reservoirs throughout the body. But I hope to end on some promising therapeutic strategies that we're utilizing to target and potentially eliminate the follicular dendritic cell network viral reservoir. So just by way of just very brief background, everybody in this group certainly knows that long-term persistence of HIV reservoirs remains the key obstacle to achieving an HIV cure or sustained antiretroviral-free viral control in people living with HIV after antiretroviral therapy is discontinued. There are many HIV reservoirs, and we operationally defined HIV reservoirs as any cell that harbors or contains replication competent or infectious virus. And the bulk of the HIV reservoirs that we know about throughout the body are infected cells, cells that bear CD4 and a co-receptor. And the principal infected cell population are CD4 positive T cells, but also macrophages, dendritic cells, and microglia can be infected by HIV. Now, these cells can reside in a variety of different what we refer to as viral states. They can be transcriptionally inactive, and we refer to those as latently infected cells, and we have tools to be able to identify those within tissues and also by molecular means. They can be transcriptionally active, what I refer to as an active viral reservoir. So these have viral RNA as well as viral DNA within the cell, and they can be fully activated or translationally active. In this case, they both contain viral RNA and viral proteins. And it's important to note that it's only this state of viral activation that can be sensed by the adaptive immune response, immune system. Then there are less studied viral reservoirs, the principal one being the follicular dendritic cell. And I'm going to talk a bit about this later in the talk. And these cells actually bind and retain infectious or potentially infectious viral particles on their cell surface for long periods of time. And we also have tools to be able to identify these cell populations. So our group is really focused on HIV cure strategies that really tackle the multiplicity of these cellular reservoirs. And we feel very strongly that the development of effective HIV cure strategies will require comprehensive understanding of the nature, the vulnerabilities, the location and extent of all viral reservoirs that persist in order to either eliminate or induce immunologic control of these viral reservoirs that are capable of reigniting systemic infection when antiretroviral therapy is discontinued. We often utilize non-human primate models as what we refer to as a window into, let me get a pointer here. As a window into infection in people that are living with infectious diseases. And we feel that non-human primate models have really provided key insights into human infectious diseases that are simply not feasible in the clinical setting. And HIV is certainly one of these principal human pathogens that is highly recapitulated both in the target cells, the pathology, the pathogenesis, and even treatment and treatment outcomes within these particular hosts. And this is because these animals have highly similar anatomy, physiology, and immune system to humans. These primates can often be infected with human or related pathogens that lead to human-like disease. And we can also perform novel prevention, therapeutic intervention, and in the context of HIV studies, cure strategies that haven't been shown to be either effective or proven to be safe in human populations. So these provide a rich model for us to understand this disease. And when we think about HIV disease, we've known for a very long time that HIV is a disease of the immune system, but there are limitations to human clinical studies. The vast majority of what we know about HIV reservoirs and the disease and the trajectory of disease is really garnered by our studies in the circulatory system in peripheral blood. And there are a lot of reasons for this. This is a highly accessible lymphoid tissue. This is a lymphoid tissue that participants are very willing to donate frequently and longitudinally. However, there were some limitations to focusing on peripheral blood studies. For example, only an estimated 0.25% to as much as 2% of the body's CD4 positive T cells actually reside at any time within the circulatory system. And importantly, many cell subsets that are infected by HIV are tissue resident cells and actually cannot be found in the circulation. So in order to understand HIV persistence and the various states of viral reservoirs throughout the body, you need to have access to the cacophony and the totality of tissues that are within an individual, which obviously is not possible in clinical studies. And so non-human primates have really provided a rich model system for us to be able to do these studies. So what I'm going to talk to you about first is some studies that we did in non-human primates using a variety of different viruses, both SIV and some SHIVs, in which we infected these animals and then we sacrificed them at various different time points before antiretroviral therapy. And then some animals were placed on antiretroviral therapy. And then we collected all of the major organ systems from these hosts. We weighed them so that we understood the mass of these organs. And then we utilized tools to be able to identify these viral reservoirs within these tissue compartments. And we've spent many years actually developing tools to very sensitively detect these viral reservoirs. And major tool that we use in my lab is called in situ hybridization, and we've really developed and optimized the RNAscope platform to where we can identify very specifically viral RNA from HIV-infected people living with HIV, as well as SIV and our non-human primates that are infected with SIV. And these arrows here point to these productively infected cells that are producing just a massive amount of viral RNA, but you can also see these individual punctate dots here within these B cell follicles, and those are actually viral particles that are trapped in that area. And then we can utilize additional methods to detect actually the single copy of integrated DNA within an infected a cell, both in infected cell lines and within an infected host. So we feel very strongly that by understanding these viral reservoirs within their native tissue compartments will help us to understand key approaches to be able to eliminate these viral reservoirs. And this just shows you some of the tools that we're able to do by utilizing these approaches. This is a singleplex, like I showed you in the previous slide, showing these individual viral particles in these infected cells, and we can multiplex this using immunofluorescence approach to be able to phenotype the cells, understand where they're localized in these various compartments, and to learn more about the properties of these cells and their activation states, and also about the cells that are surrounding and interacting with these infected cells. We've also, in the last several years, begun to develop tools that allow us to look and to understand the dynamics of these cells within the volume of these tissues which they reside. And I'm not going to talk about this any further today, but I just wanted to highlight that when we look at serial sections of lymph nodes, for example, we can build what we refer to as these 3D atlases of viral RNA positive cells. And we can quantify the extent of the viral RNA that they're expressing, shown here on this heat map. And you can see in this context, we see low numbers of these active viral reservoirs in some of these tissue compartments after long-term suppressive antiretroviral therapy. But just 12 days after release of antiretroviral therapy, these expand into a massive population that are extended throughout the volume of these tissues. So in the study that we performed, in which we collected all of the major organ systems And we looked for both active and latent viral reservoirs throughout these tissue compartments. The first takeaway I want to show you is that we found virus in all of the major organ systems, both before and during antiretroviral therapy. And when we quantify the number of these infected cells per gram of tissue and then extrapolate that into a total population within these organ systems based on the mass of the organ, we then can begin to understand the total proportion that these organ systems contribute to the total body burden of these infected cells within a living host. And what we found was that these active viral reservoirs, and not showing you here, but also total viral reservoirs, really predominated within lymphoid tissues. And when we quantify that, greater than 99% of all the viral RNA positive cells resided within lymph nodes, gut, spleen, and mucosal associated lymphoid tissues. So while we're able to see infected cells within the brain, the liver, the heart, and other organ systems, they actually contributed very minorly to the total body burden. When we looked at this after 26 weeks of antiretroviral therapy, the abundance of the viral reservoirs did decrease, but the proportions actually changed quite substantially, in which the gut became the dominant source of viral RNA positive cells throughout the body in this case. And when we look at this in all of these various organ systems, so now we're looking at viral RNA positive cells per gram of tissue. The black bars are the levels prior to antiretroviral therapy. The blue bars are the quantity after antiretroviral therapy. We see a couple of different themes. We see some tissues in which we see multiple log reductions of these active viral reservoirs during antiretroviral therapy. We see other tissues, such as the gut, in which we see only several fold reductions. In this case, only a 3 to 4-fold reduction. And then, such as the central nervous system, particularly the brain, we actually don't see a reduction in the number of viral RNA positive cells. So even though quantitatively these represent a very small fraction of the total infected cells throughout the body, it doesn't appear that antiretroviral therapy actually particularly impacts the number of these active reservoirs within these sites.
Jake, may I ask you a question?
You can, yeah.
This is Michael. I just wanted to ask, so that looks like the brain increased slightly. Is that reading that correct? Would that look like, how do you interpret that rise? What is going on there? I'm not understanding.
Yeah, so that is a great question, Michael. So keep in mind that these are the means of individual animals. So all of these animals had to be sacrificed. So this isn't a longitudinal study.
Okay.
This is a cross-sectional study. And so these were not statistically significantly different from each other. And we're doing more and more of these animals over time. So we'll get a better sense of this, I think, over time and with longer-term therapies as well.
Okay. I was just wondering, like, I know neurological conditions with HIV are a big issue. And when I see something like that, it just makes me think about a connection to that.
Yeah, we're thinking about that a lot, actually. And we definitely think that inflammation driven by persistent active viral reservoirs, as well as persistent systemic inflammation, actually may be a significant contributing factor to neural inflammation and potential HIV-associated neurocognitive disorders as well.
Thank you.
Yeah.
So Jake, this is Marc Wagner. I have just several questions. So I'm assuming that these animals, even though the tissue viral loads are not going undetectable, but their blood viral loads are undetectable. Am I correct to understand that?
That is correct. So by 26 weeks, these were under 15 copies of viral RNA per mil of plasma.
Okay. Now I know you've looked at various tissues, but have you looked at the brain even at this point where they're on antiretroviral therapy and even without, and discerned where in the brain this virus replication is taking place and what cells are actually lighting up for both the DNA and the RNA that you're looking for.
Yeah, we actually published a paper with Melissa Churchill last year or the year before. It's in PLOS Pathogens, and we have a more extensive analysis. We focused on the frontal lobe in that particular paper, and we found it both in the gray and white matter. So we didn't particularly see, enrichment, at least in the frontal lobe, and they predominated in microglia.
Okay.
Yeah.
Thank you.
You're welcome. So one of the things that we're able to do with these data is actually quantify the total number of infected cells within these different tissue compartments. And from that, the total HIV viral burden of total DNA positive cells. And then when we take that number and we divide it by 2 to 5%, which is the estimate of the intact or replication competent viruses during antiretroviral therapy, we estimate between 10 to the 7th to 10 to the 8th replication competent viral reservoirs in these tissue compartments persist throughout the body. And this is really important because work from Bob Silicano's lab recently demonstrated that this rebound competent or replication competent viral reservoir does not appear to decay over time. And so we really need to develop therapeutic approaches that actually specifically target these rebound competent, replication competent viral reservoirs.
All right. And this is Marc again. If I can just highlight that is really important because you're talking about tens to hundreds of millions of cells that are being represented in that reservoir. So we know that all you need is one to do it, but here we have tens to hundreds of millions by your estimation. Is that correct?
That is correct. I mean, it's a daunting number, but I think knowing the extent of this particular viral reservoir is important in order to understand what the delta is necessary in order to have, effective elimination and or effective control of these viral reservoirs.
Absolutely. Thank you.
Yeah.
Jake, I was going to wait till the end, but it might be best to ask it now. Could you go back to your last slide, please?
Yeah.
So, you have these different compartments. We can see the ones that are the most affected. And, I always wonder, if suppose somebody has compromised kidneys or something, it may not take as much HIV to affect that. I don't know. I wonder sometimes, I think about this like cancer, which might be all wrong, but do the different compartments react differently? If we're going to if we find some intervention in the future, are we going to need different interventions for different compartments? You know what I'm getting at? It's like,
no, I do.
Is that relevant, or am I off the money here?
No, I'll maybe answer this shorter than I probably normally would, but I think there are multiple aspects to that question, Lynda. The first is drug penetration. Therapeutic penetration that can actually target effectively, viral reservoirs within those tissue compartments. That's the first thing that we don't know. I think the second is effective immune responses that could potentially mediate control and or elimination within those various tissue compartments. And I think that will be different. One of the things that we have learned over the years is, you take macrophages, for example. We tend to think of them as a single kind of population, but they're vastly different in each of these various tissue compartments. A comfort cell is very different from a gut macrophage, which is very different from a lymph node macrophage. And so I think that the approaches that we take are going to have to take into account the heterogeneity of these viral reservoirs throughout the body. And I think, the brain is one that's very high on my mind, because drug penetration and the effectiveness and the side effects of potential therapies are going to be a big challenge.
So it's more like pharmacology or pharmacodynamics than the actual intervention itself.
I think it's both.
Okay.
And you have to also layer on the immune control component as well.
Okay, which is a huge.
Which
All
is
right,
big.
great. Thanks.
We had another question I just wanted to ask, since it's kind of related. Someone wanted to know, with such a large concentration in the gut, are we talking specifically about lymph tissue in the gut or other gut tissues?
Yeah, so when we quantified the total population within the gut, we quantified throughout the entire gut, from mucosa all the way through to the muscularis. And what we found is that most of the virus that persisted in these secondary and tertiary lymphoid tissues, so the mucosa and the follicular aggregates and Peyer's patches. You do find them in other regions, but they're much rarer in those sites. And what I'm not going to be able to show today, just because of time, we're spending a lot of time in trying to understand why this distribution is the way that it is. And what we have found is that the distribution of these viral reservoirs really tracks beautifully to the CD4 positive, CD4 T cell population size. So we see CD4 positive T cells in all of these tissues, even in the brain. But where we see the 99% of these reservoirs residing are in these tissues that have just massive populations of CD4 positive T cells.
And this is a related question. Someone was saying that they'd be interested in seeing if liver concentrations go up for those people who have hepatitis infections.
That's a really good question. I could only speculate. I don't know. Yeah.
Great. Thanks. Go ahead.
So what I hope you kind of saw from this previous picture is this large abundance of virus in these particular locations. This is the spleen, and this is a lymph node. And this is what I refer to as this FDC network, this virus that's trapped on this network. And you can see these individual viral particles that are trapped on these dendritic cells that are not classical dendritic cells, but they bind and retain these viral particles for long periods of time. And we've known this particular cell and these environments have been a source of viral storage for quite some time, for over 30 plus years. And these are just old photomicrographs from multiple publications that have just shown the large abundance of this virus that's localized on the follicular dendritic cells of these secondary lymphoid follicles, and also within SIV infected animals. And so we asked the question, if we were to quantify the total amount of the virus that's bound to the FDC compared to the total amount of viral RNA that's contained within productively infected cells in a chronically infected animal, wanted to know what the difference in the tissue viral burden was. And so we are able to quantify the productively infected cells within these tissues. We're able to quantify the viral RNA on the FDCs. And what we find is that between 90 and 99% of all the viral RNA within lymph nodes is actually trapped and retained on FDCs at any given time during chronic infection. And so when we quantified that across multiple secondary lymphoid tissues, so these are different mesenteric and colonic lymph nodes and peripheral lymph nodes, we can see that this network is actually seeded relatively early. Within 14 days, it's maintained at this high level throughout chronic infection. And when we estimate based on the mass of these lymph nodes in this infected host, we find that there are greater than 10 to the 10th viral particles that are actually trapped on the FDC prior to antiretroviral therapy. But we know that this actually changes with antiretroviral therapy. So how does this look? So this is an example of an animal that had nearly 7 times 10 to the 6 copies of viral RNA per mil of plasma. This is what this animal's lymph nodes look like. Lots of deposited virus on the FDC. You see infected cells within this region. After 26 weeks of antiretroviral therapy, this animal is below the limit of the clinical assays to detect it. It has a very small amount of viral RNA still found within the tissue. So you take the tissues out and you homogenize it and you quantify the RNA. You have about 5 copies per 10 to the 5th cells. But when we look in the B cell follicles, we can still see these rare but identifiable viral particles that are present there. And this is another example of an animal below the limit of detection during antiretroviral therapy, has a very low copy number when you homogenize and quantify it, but we can see that there's still virus that's retained there. And when we quantify this, we see that between the chronic state and 26 weeks of therapy, we have a nearly 3 log reduction of virus. So this is a substantial reduction in virus in this location. And this is consistent with what Ashley Haas's group showed nearly 30 years ago, in which they saw about a 3.4 log reduction in people living with HIV that were on antiretroviral therapy for 1 year. So this animal model models what we see in humans. But if you take this quantity that we see prior to antiretroviral therapy and you apply a 3 log reduction to it, and then you make these quantitative estimates, you still are left with another large daunting number of approximately 10 to the 7th viral particles that are trapped or potentially trapped on FDCs during long-term progressive therapy. And lots of work have been done in trying to understand how this virus is trapped and retained on these follicular dendritic cells, and is this virus infectious? And this paper that was done by Michael Carroll's group about a decade ago actually demonstrated that if you take FDCs out of secondary lymphoid tissues from individuals that have been on long-term suppressive therapy from anywhere from 1 to 20 years, and you ask the question, can infectious virus particles reignite new rounds of infection ex vivo? The answer is yes. So the FDC can retain this virus in an infectious state. But we know that it's trapped and retained on the surface via these complement receptors, in particular the CD21 complement receptor 2 molecule. And so we wanted to ask the question, can we treat animals, in this case, with an antibody to CD21, and would that attenuate the establishment of this FDC HIV reservoir within these B cell follicles? So we did a proof of principle study in which we took 5 animals and we injected them with a clinical asset from our partners from ViiV GSK, Rick Dunham and Heather Madsen, in collaboration with Michael Carroll. And we injected them with an anti-CD21 antibody every 2 weeks, infected them, and then let them go for 8 weeks. We took serial lymph node biopsies, both peripheral and mesenteric, and then sacrificed the animals at 8 weeks. And we compared them to animals that received an irrelevant antibody at the same concentration and asked the question, is this sufficient to prevent the deposition of this virus on this follicular dendritic cell network? And the answer is yes. So when we look as early as 14 days after infection, so after 1 injection of antibody, here's the controls. We see a substantial amount of virus that's already deposited on the FDC network, and we can see a substantial reduction. And then when we look over time at 28 days and 56 days or 8 weeks, we see that this persists. So this one treatment alone was able to prevent or substantially attenuate the deposition of virus within these B cell follicles. And when we quantify that in these animals over time, we can see nearly a 20-fold reduction in the amount of virus that's deposited on these FDCs compared to our control animals. Importantly, when we look at multiple lymphoid tissues, axillary lymph node, mesenteric lymph node, spleen, and gut-associated secondary lymphoid tissues, we can see that this effect actually extends into these areas. Slightly less effective within the gut, and this is not inconsistent with what we've seen with other antibody treatments. The penetration in the gut seems to be less effective, but nonetheless, we see that we're able to substantially reduce this. Now, this is a proof of principle study. We're never going to be able to treat individuals prior to infection. And so we performed another study in which we infected animals, let them go for 6 weeks. They established this FDC network, and then we treated them with anti-CD21 antibody or control antibody at the time that we treated them with antiretroviral therapy, and asked the question, does this accelerate the decay of this virus or lead to the shedding of this viral reservoir above what we see with antiretroviral therapy alone? And the take-home message from this is that when we look at this over a 12-week period of time, we see that this anti-CD21 treatment actually accelerated SIV decay on the FDCs about 50% faster than ART alone. So what I hope that I was able to show in this short period of time today is that before and during antiretroviral therapy, that viral reservoirs are distributed throughout the body, but predominate in lymphoid tissues, both the gut and lymph nodes and spleen. That active viral reservoirs decrease substantially in most tissues, but not in all tissues. That active viral reservoirs remain detectable in all tissue compartments during antiretroviral therapy, but most of quantitatively reside within the gut in these infected hosts. And that the follicular dendritic cell network is an important uninfected viral reservoir that we really need to think about and need to address during our HIV cure strategies. And we're exploring the promising therapeutic strategy of this anti-CD21 as a potential component in a cure-related strategy that most certainly will include a multiplicity of different potential treatments to target all of the viral reservoirs within an infected host. This was work that really spanned over a decade in my group, and is still work that's ongoing. And there's been a large array of individuals, and they're listed here. A lot of the work that was done with the anti-CD21 was led by very talented postdoc Chi Chan. Kathleen Busman-Sahai is my scientific program manager in my group and a research associate professor, and she's been with me for a long time and has been involved in all of these studies. And I also want to thank the funding for this, and thank you for your attention, and love to have more discussion.
Great, thank you.
Great.
Really great. So go for it, Michael.
No, go ahead. I'm going to take care of some things first here, and then I'll get to the questions in the chat. But if you want to ask something first, go ahead.
No, I was going to wait.
Okay. There's lots of questions that came in. So I'm going to try to do these. I'll probably just go backwards through what we have here. So someone wanted to know, did we see common decreases in viral reservoirs among different subjects on ART? Did most have decreases in the same reservoirs, or was it different for each one?
In these non-human primate studies, we saw a similar decline in our animals. So we saw between a 2 and 2.5 log reduction in lymph node for active viral reservoirs in all of our animals, and we saw only a 2 to fivefold reduction. So only as much as a half a log reduction within the gut.
Mm.
Importantly, when we looked at the total viral reservoir decline, so all viral DNA positive cells, we only saw about a half log reduction, which is actually consistent with what we see in people living with HIV. The total DNA viral reservoir only declines anywhere from a half to as much as 1.5 logs over the first 6 months of treatment. And then it really kind of tapers off, and it's a very slow decline after that point. And so, I think that antiretroviral therapy is, a remarkable tool, and it's been a life-saving therapy, but it is not a therapy that is going to effectively eliminate viral reservoirs.
And so another question is, does reducing systemic inflammation increase the concentration of infected cells, or does it reduce the load?
It's an excellent question. We hypothesize that persistent systemic inflammation is a significant contributor to viral persistence, and it does so through a variety of different ways. We think that systemic inflammation leads to potential clonal expansion of infected reservoirs, and that those clonally expanded cells are the significant means for persistence. And we're looking at this. We have an ongoing study in which we are looking at 2 anti-inflammatory therapeutics during antiretroviral therapy in non-human primates. One is an anti-TNF inhibitor, and another is an anti-inflammatory that's under clinical evaluation with GSK that also targets the TNF pathway. And so our hypothesis is, if we can reduce systemic inflammation, we can restore gut homeostasis and the epithelial barrier within the GI, and that will lead to a reduction of both active and total viral reservoirs within that compartment.
Someone wanted to piggyback off of that question and wanted to know, does this effect depend upon the reservoir type?
That's a really good question. We don't know. Yeah, we don't know.
All right, let's see. So next question. Now wondering, what's the general observed margin of error when comparing SIV pathogenesis to HIV pathogenesis?
Yeah, there are differences. I think the SIV or SIV models, and depending on which virus you use, which species, it's an accelerated model, and so animals untreated will typically progress to end-stage events within a year and a half on average. We obviously know that that's longer in humans. But the types of pathology that we see in HIV, in people living with HIV, is recapitulated in the non-human primate models. We see lymphoid tissue fibrosis. We see persistent GI dysfunction and colitis. We see the CD4 T cell decline is also very similar. We see blood-brain barrier dysfunction in SIV-infected non-human primates. So the pathology is actually remarkably similar, but it's slightly more accelerated, I think, in the SIV-infected hosts.
And was bone marrow included in any of these reservoirs that you mentioned?
Yeah, it's a great question. We have looked at bone marrow. We did not in our total body viral burden work. But we have looked at bone marrow, and we do see infected viral reservoirs within the bone marrow, but it's remarkably low. And we've struggled to, I think, effectively phenotype them. They've in all of our attempts to phenotype those infected cells, they're always CD4 positive T cells, but we don't know at what stage those CD4 positive T cells are in. We haven't been able to dissect that out any further.
Mm-hmm. And was the sex of the primates taken into account?
It's an excellent question. It's something that we struggle with quite a bit because non-human primate studies are very expensive. Having sex-matched studies is also difficult. And the majority of the studies that I showed you were in male rhesus macaques. Some of the early work that we did with the total body distribution is in both male and female. So we definitely know that there are sex differences, but we've struggled to try to tease those out, get enough a high enough N to be able to definitively see what those are.
Okay, here's a related question too. Did you look at the viral reservoir in female breast tissue?
Great question. No, we did not. No, we did
There's
not.
a good one for you, Jake.
Um,
Let me ask you
okay.
about the monkeys. I mean, how about postmenopausal monkeys? I mean, is it easier to find females in that demographic to get, I should say, not find?
Yeah, so all of these are purpose-bred animals. And the older an animal gets, the more challenging it becomes to put that animal on study. So no, we have not looked at aged non-human primates. There are some aged colonies, but they're quite difficult to actually maintain. A little bit easier for females actually because they can stay in the breeding colony for longer. Males have to be removed because they're aggressive and they cause problems in the social structure. And when you have to remove them, you put them in a cage, and an animal that's in a cage for a long period of time just doesn't do as well as when they're in, their social communities or very socially structured animals. So that's a really good question, and I have to think about a really good way of trying to address that.
And Anna wanted to know about, could you say more about the viral reservoir in the uterus?
No, I can't. It's there. We see it in vaginal mucosa. We see it in cervix. You can see it. We see it all the way up through into the ovaries to various extents. They tend to be more abundant within these tertiary lymphoid tissues that emerge within the mucosa of the female genital tract, mostly within the cervix and the vagina, but you do see infected cells within the uterus. And there in our hands are almost always CD4 positive T cells.
And someone also asked, what are your thoughts on the brain reservoir?
You think about it all the time, right?
Yeah, we think about this a lot. In fact, we just put a grant in with Melissa Churchill. We have a study we just completed in which we took animals to 72 weeks of antiretroviral therapy. We treated them early at day 9, and then when we sacrificed them, we actually took all of the brain compartments. We took all of the lobes. We took the cerebellum. We took the midbrain, we took the hippocampus, and we want to do a comprehensive evaluation of the viral reservoirs throughout each of those compartments. And then we also had 2 groups of animals that came off of antiretroviral therapy, and we sacrificed them at days 5 and 7. So we also want to know what contribution do the viral reservoirs within the brain potentially contribute to off our viral rebound when antiretroviral therapy is removed. I can't tell you the work that we've currently done. Most of the viral reservoirs that persist in the brain of SIV infected rhesus macaques or microglia, they can we find them perivascularly. We find them deep into the gray and white matter in the parenchyma of the brain. We see persistent neuroinflammation throughout the brain. And it doesn't necessarily track with where these active viral reservoirs are. And so we think that systemic inflammation is contributing to the persistent neuroinflammation within the brain. And we also see persistent blood-brain barrier defects in animals that are on long-term suppressive therapy. So that's kind of what I know so far, but more to come.
Are there any ethnicity or race parallel characteristics within simians that could help us predict possible differences in how medicines are metabolized in different human ethnic groups? If so, are any of those factors examined?
That is such a good question. Not to my knowledge. Not to my knowledge. I think probably not to my knowledge. There are multiple different species, and there are species differences. Pigtail macaques, for example, are, yearly breeders, yearly cyclers, whereas rhesus macaques are seasonal. You have differences in Indian origin rhesus macaques versus Chinese origin rhesus macaques. So there are some differences. Whether that would equate to race differences, I don't know.
Here's another question. Faith asks, with aging, ratio or number of infected cells changes in reservoirs? And is there any change in the leading reservoirs ranking, which includes the biggest ratio or number of infected cells?
Yeah, another great question. We don't know. Most of these studies that we have done have been in animals between the ages of 4 and 10. So these would be classified as adult rhesus macaques, but they would not be classified as aged. We do know that the immune system dramatically changes with age. We do know that lymph nodes, for example, hyalinize. They actually develop fibrosis over time in humans, and we suspect that also happens in macaques. We know that thymus function substantially decreases during adulthood. So I suspect that those also occur in non-human primates, but we just have not looked at an aged population.
The blood-brain barrier defects, were these defects resultant of ARV use or the long-term HIV effects, or both?
Yeah, the actual the blood-brain barrier defects occurred prior to antiretroviral therapy initiation and persisted after. So we don't think that they're associated with the drugs themselves. And the strongest correlation that we found to the blood-brain barrier defects actually was GI inflammation. So we're really interested in understanding this gut-brain axis and the effects of persistent GI epithelial damage and microbial translocation and inflammation resulting from persistent microbial translocation and the impact on the blood-brain barrier. We in this paper we published with Melissa Churchill, there's a group of animals that is in that paper that are completely SIV uninfected. And we developed a chronic colitis model in non-human primates to try to understand how that gut dysfunction leads to systemic inflammation. Well, when we looked at the blood at neuroinflammation and blood-brain barrier of those SIV uninfected animals that had chronic colitis, they looked very similar to our SIV infected animals. And so we feel the data suggests that GI related damage and inflammation is a significant contributor to neuroinflammation and the effects that we see in the CNS.
So Patricia asks a good question. In the known cases of cure, have there been ways to check on whether the FDCs are still present, knowing that it would be invasive to do biopsies and we've poked these folks so much already?
Short answer to that is I think in a few cases, lymph nodes have been taken, and we've certainly had samples from a handful of those participants, and we have not seen persistent virus on the limited sections that we've looked at. You have to keep in mind you're looking at a very thin section of a lymph node, and so in order to rule out something as not being there, you have to do a much deeper survey. But we haven't seen that, and we think even if there were viruses there, they most certainly, we suspect they would be CCR5 tropic viruses, and in individuals that are reconstituted with the delta 32 hematopoietic stem cells, they wouldn't lead to new rounds of infection even if that virus did persist.
Is the effectiveness of HIV drug types tested, like newer drugs versus older drugs affecting the reservoirs and the types of drugs? Or instead of triple therapy, how about double therapy? Was that tested?
Yeah, excellent question. We have not. I know Greg Delpret and Jeff Lisson have done a lot with antiretroviral therapy treatment in non-human primates, and they've certainly looked at things from monotherapy up to triple therapy. They've looked at triple therapy going down to monotherapy or to dual therapy. But that's a really good question, and we are putting in a grant with Angela Kashuba's group, who really has kind of pioneered imaging tools to be able to quantify antiretroviral and their distribution and penetration within tissues themselves. And we have developed approaches to be able to overlay our viral measures with their drug measures. And we're putting in a grant actually to look at these long-acting antiretrovirals in the non-human primates to see if distribution and penetration and concentration are fundamentally different, and how those change at different times of ART initiation. So one of our hypotheses is that individuals that start therapy later in disease have a higher extent of pathology in their lymph nodes, more fibrosis, more cellular dysfunction, and likely can lead to poorer antiretroviral distribution within the tissue compartments in which these viral reservoirs reside. And so we want to look to see if those relationships actually exist, and if long-acting regimens actually are better at penetrating into these tissues.
Another question was, does what you feed the babies affect the development of colitis in adults and the neural inflammation?
We haven't done infant studies, so I don't know that. Certainly diet certainly can affect the microbiota and GI development. But all of the studies that we have done in in our chronic colitis model have been in adolescent to young adult macaques.
Okay. That's all the questions in the chat.
Okay.
Wow,
I
Jake,
have
you've got more
a few-
questions than anybody.
okay, I'll take,
Come on.
I have a few. I'm assuming you want to go to clinical trial eventually with the anti-CD21 antibody, and I'm assuming that has to be given by IV infusion. And I had several. Your one slide actually showed it looked like the control IgG also seemed to decrease the levels of the virus as well. And do you see cytokine changes? And also, do you think there'll be antibodies raised against the antibody, the anti-CD21, that maybe will make it not so effective once you go in the clinical trial?
Yeah, lots of questions there. Let me try to dissect those. Yeah, we certainly hope to, progress this in clinical trials. It would be a pretty challenging clinical trial because the only way to actually test the effectiveness of the actual the treatment itself is to monitor FDC deposited virus, and that would require lymph node biopsies, and it's a challenge. So that's first thing. The second is that our study that we did sub-Q injection. So these antibodies can be quite effective with a sub-Q injection. They're often given through IV infusion in the clinic, but I think that they can that the administration route can be altered. The control IgG that you're referring to actually was the ART-only control. So those animals were placed on antiretroviral therapy and given a control antibody. So the decay that you saw is almost certainly due to the antiretroviral therapy shutting off, viral production. And so that source of deposition does decrease with antiretroviral therapy. There's no doubt. And then the third thing is that the one thing that we're really thinking about with this particular therapy is, and I didn't have time to show you, but we also saw that this therapy led to less infection in the T follicular helper cells. And the T follicular helper cells is, we think, an important population of infected cells that persists in people that live with HIV. And we think that the relationship between the virus that's trapped on the FDC infecting those T follicular helper cells, and then those infected cells seeding more virus on the FDC is an important viral dynamic process. And so we would like to explore the potential of using an anti-CD21 in analytical treatment interruptions as a means to prevent the reestablishment of FDC reservoir and potentially limit T follicular helper cell infection in cure-related strategies. So we haven't really explored this further. We're in the process of writing this paper, and then we'll have further discussions with our pharma partners, and hopefully further discussions with the NIH to potentially fund this. But this, we think, could be a potential means to add more safety to cure-related clinical trials down the road.
Okay. I have another question too. About 8% of the human genome DNA is remnants of old retroviruses. And, what goes along with that is you'll see gag pull an envelope. And, I don't know if, and that does create a problem when you're trying to go with probes that will, identify the virus. So how do you think in the primates that you'll have a similar issue as you might have with humans? And
yes.
quantification and looking at, given that these old viruses no longer producing virus, but have DNA that could cross-react.
Yeah, so we have not seen that. The approach that we utilize, the RNA scope, is quite sequence specific. The probes only have a very modest ability to have sequence divergence between the probe and the target sequence. We haven't seen in any of our SIV uninfected animals potential cross-reactivity of our either DNA or RNA targeting probes to a significant extent. I mean, with any assay, you have a limit of detection, but our limit of detection is just under 1 in a million total cells. And so when we see an infected cell and we see the threshold of viral RNA within those infected cells, we're pretty confident that we're picking up the infected cell and not some remnant sequence.
Okay.
There was a question about what do you think about the effect, what the effect of the newer drugs, like the injectable therapies that last for a lot longer? What would they have on this type of studies that you're doing? It might be too early for you to comment on that, but
no, it's a great question, and I'm excited to hopefully come back in the next couple of years and answer that question. If we can get this grant funded, we will have a much better sense on that. I'm really hopeful that the long-acting regimens have greater tissue penetration, potentially have a stronger impact on, for example, the CNS viral reservoir. But some of the early studies with previous regimens, when we looked with Angela's group at antiretroviral distribution in the brain, it's almost it's very low. It's very centered around the perivascular space, but does not seem to disseminate into the parenchyma of the brain to therapeutic levels. And so love to be able to see that actually changes with some of these new regimens and new approaches.
And this isn't a question, it's just a comment, but I just wanted to share it with you live here. So you have to start somewhere, they said. Being someone living with HIV, I appreciate everyone associated with this type of work. Very informative and exciting to follow this.
Thank you.
I just want to let you know that.
Cool. Okay, so Jake, let me ask you a couple science questions, and then I'll get back to the more community aspect part of this. So, in other words, I know that you're not looking at immune functions particularly, but is there a way for you to tease out if there may be some sort of differences in different animals, like there might be in different people, that account for different behavior of the virus?
Mm-hmm. Yeah, so one of the things that my group is really focused on is trying to understand in much more comprehensive way the actual cells, the immune cells that interact with these viral reservoirs. We think that what we refer to as the cellular neighborhoods, or those cells that are in closest proximity with those infected cells, play an important role in either the control and elimination of that infected cell or the persistence. And what we have found is that there is an enrichment of immunosuppressive environment around these infected cells. And what that means is that either the infected cell itself, or just by virtue of the environment in which these cells persist, is leading to an environment that is going to be more difficult for the adaptive immune system to be functional and potentially clear that infected cell. And so by understanding these environments that these infected cells are residing in, we can then think about therapeutic interventions that could block that. For example, work that Rafik Sekali and Mirko Peridini are doing really stem from some of our early work that showed that these cells tended to reside in an IL-10 rich environment. And now we're looking at means to block IL-10 in addition to enhancing the immune function through an anti-PD-1, for example, and to look at the outcome in these non-human primate scenarios, these models. And what we've been able to show is that when you take an animal off of antiretroviral therapy and you treat with anti-IL-10 and anti-PD-1, both before and after you remove an animal from antiretroviral therapy, we can actually extend viral control for much longer than the untreated scenario. So we think that understanding these environments and those cells that are interacting will be actually pretty key to that immune control component.
I didn't know you were working on that. That's wonderful. Michael, do you want to take Lewis's question that relates to this?
So related to what you were just saying, Lewis asked, so you're saying that because of the cellular neighborhood is constantly activated, it leaves the adaptive portion of the immune system depleted or otherwise less able to respond to control HIV in the reservoirs they surround?
Yeah, it's probably a little bit more complicated than that. I mean, there are two things to maybe think about. First of all, cells are not static, right? They're always in a state of motion and movement. And so cells are likely going in and out of states of inflammation and in states of what I refer to as immunoregulatory environments. And what we have found is that when we take these tissues out of the hosts at any given moment in time, and we look at what's the theme, if you will, or a consistent relationship or immune responses that are happening around those infected cells, we see both pro-inflammatory but also anti-inflammatory signals that are within those environments. We see a lot of T regulatory cells, and we see a lot of pro-inflammatory mediators. So there's this yin and yang, I think, always going on with these viral reservoirs. However, if you look at cells that are just 50 microns or 100 microns away from that infected cell and ask the question, so they're in the same environment but not close to a productively infected cells, we don't see that same relationship. We don't see that they have these anti-inflammatory mediators that are there. So we think that the immune response is responding to those infected cells, particularly the active infected cells, right? The ones that the host can actually sense. And there's both a pro and a counter-regulatory response to that infected cell. And we think that counter-regulatory response is probably limiting the host's ability to actually clear those cells.
Oh, and so you've talked a lot about inflammation. I mean, it's been a theme throughout. It sounds like we're going to need some sort of an anti-inflammatory component to every regimen. I mean, is that something that you think about?
It's something I think about all the time. It's something that we're studying, actually. I think that there are. I think that people living with HIV that are on long-term suppressive therapy, I think can have improved lives if we're able to actually address this persistent and systemic inflammation. We know that it is associated with increased comorbidities, right? We know that it's associated with impacting life expectancy. We know that this is, a significant problem. But I think that, and I think that we can address that with adjunctive therapies that actually address the actual source of that systemic inflammation. But I don't think that we have great tools yet to know how to do that. And that's where these non-human primate studies, we hope, will lead to better tools to address. We think the GI is the major source of systemic inflammation. We think, based on studies we've done with people that live with HIV, as well as our non-human primate studies, that we see persistent dysfunction in the epithelial barrier of the GI. We see microbial translocation persist in individuals and primates that are on long-term suppressive therapy. And that's not normal. We don't see that in age-matched controls. And so we have to be able to address the source. And if you can't address the source, I don't know, it's like putting a Band-Aid on a dam, I think. You have to actually stop the leaks.
So I don't know that these are so much questions as, well, let me do it this way. So you work in the preclinical world, and it sounds like you're moving a little bit into the pharmacology world of human beings. So have you thought about working with clinical trialists to deal, to somehow reach the questions and answers that you're looking for? One thing I might suggest that came to mind is the Last Gift program, and that in other words, people that volunteer their bodies after they're gone, because there's a lot of stuff, as you said right in the beginning, that you cannot do, that you do now with primates that you can't do with people. So I just wonder if you might have thought about that sort of a nexus.
Yeah. So we have been involved in a number of clinical studies. We've participated in a handful of ACTG clinical trials. Telmisartan was one of them. We've been involved. We've collaborated with a variety of individuals that have done cure-related studies. The challenge is the types of analyses that my group does, requires, extensive tissue collection. We wouldn't know the extent of HIV distribution, throughout the body if we weren't able to actually collect the large array of samples that we do in the non-human primates. And we have had conversations with folks in the Last Gift cohort, but I think that funding limitations, I think sample differences have probably limited that to some extent. The challenge with kind of the Last Gift cohort, as I think truly remarkable as that is, when an individual passes away, the body begins to deteriorate right away. And so we've seen, and we've gotten samples from some of these autopsy studies, that it becomes a challenge to actually effectively quantify these viruses as that RNA is beginning to degrade. So there are challenges, and we're certainly open to engaging more in this capacity, though.
Yeah, I mean, it's just that your work is so practical, you know, I mean, compared to some of the stuff that we see, it's just, it's such a natural progression, but certainly there are lots of problems. And I just wanted to reiterate, a lot of the things and a lot of the questions and a lot of the points that people brought up about women, younger women or older women, the differences there, racial differences. So, I mean, those things are always on our minds, and you know, you may not be in a position to do that now, but I mean, we just urge you to think about that all the time. I mean, you know, I mean, more than 50% of the people with HIV are women. Certainly the same thing with people that are chronically infected. And everybody wants to get. It reminds me of the early days when, okay, this was going to look better in naive people, so that's what we're going to do. Well, then, you know, and newly people with that are just getting HIV are in better shape. So, you know, I mean, it's the same thing with a different pair of pants, you know, with a different outfit. So we have to worry about people that have chronic infection too. But anyway, this has just been really wonderful. We've had some great questions and very interesting sorts of from answers from you I mean, across the gamut of work that you're doing and that's we're thinking about. So let's see, we've got a couple of minutes. Does anybody have some other questions? Michael, you've been good.
I'm trying to look through it all. So let's see. Someone was asking about, are there studies or guidelines on gut health, diet, and inflammation that would help lower folks' inflammation?
Yeah, that's a great question. I don't know.
Yeah, there are.
I'm sure there are.
Find a link to it. Yes, there are. I'm just trying to like find where, you know, like it's basically searching online. But yes, the answer is yes. Diet is a big part of it, but let me find it. Here you go.
While you're looking, Michael, go ahead.
I was going to just say, I'm going to put the link in the chat for people who are interested in that from Pause magazine.
I mean, I was wondering in the gut, is it just because there are more receptors there, or is there more of a brain gut sort of interaction that causes this?
Yeah, so we actually don't fully appreciate the gut brain axis. At least I don't fully understand the gut brain axis, but we think that the dysfunction to the GI actually happens very early. Prior to infection, there is a large population of effector memory CD4 positive T cells that are localized within the lamina propria of the GI throughout the entire small to large bowel, even up through, you know, the oral mucosa all the way down to the anus. You have these very large populations of effector memory CD4 positive T cells, and they are a principal target cell for HIV early in infection. And those cells are actually depleted quite extensively within the first 4 weeks of infection. And reconstitution of those cells is very limited after long-term antiretroviral therapy. In fact, we see, less than 50% of the CD4 positive T cells actually will reconstitute in the lamina propria after long-term suppressive therapy. And so in those populations of cells, IL-17 producing Th17 cells are very important for the maintenance of homeostasis of the epithelial barrier. And so we see gut barrier dysfunctions very early in infection that don't fully restore during antiretroviral therapy. And so until you're able to effectively restore that gut barrier, you're going to have luminal bacterial or luminal bugs always in contact with the host, which is highly inflammatory. And we think that that's probably one of the major sources of persistent inflammation in people living with HIV.
And, I mean, we've got a minute here. I wasn't going to go into this because I don't fully get it, but what's different about the FDCs? Why are they uninfected, and what can we learn from that?
Yeah, so follicular dendritic cells, I think the fact that they were called dendritic cells at all is a bit unfortunate, because folks tend to think that a dendritic cell is an antigen presenting cell, or a classic dendritic cell is an antigen presenting cell. But this is a stromal population, a stromal cell that resides only in the B cell follicles of secondary lymphoid structures. And its primary purpose is to induce humoral immune responses and B cell memory. And it does this by providing signals to B cells and T follicular helper cells, but it also does this by retaining antigen and periodically presenting that or releasing that so B cells can become activated and stimulated. And so the reason, for example, you only need a tetanus shot every 10 years is because presumably some of these antigens are retained in these structures, and they are presented to B cells so that they can proliferate and maintain that long-term humoral memory. And what happens in HIV is that process, that normal biology of binding and retaining antigen is saturated by this enormous amount of virus that's being produced within these lymphoid structures. And so you have viral particles that have antibody attached to them, have complement byproducts attached to them, that now are binding to these FDCs, and binding at very high levels. And so it's actually just performing its normal biologic function is the way that I see it.
Cool. All right. Well, anybody else before we let Jake go? This has been amazing. It's really been one of the best ones we've had so far on this, Jake. Thanks so much for taking the time to be with us. Thank you, Marc, for your great questions. And for everybody who attended. I hope you learned something, and I hope you enjoyed it. I have put a link in the chat for the DAIDS Community Advisory Board. So if you'd like to get involved with activism, from vaccines to cure to clinical trials, whatever, prevention, vaccines, you'll find a community advisory board within that link that you can get in touch with. So it's not over, and we could use your help. So thank you again, Jake, Michael, for being always old faithful. And we'll see you all next time. For the next time, we're going to be talking about new assays with Matthias Lichterfeld. That's going to be so interesting, because it would be great to get rid of these analytical treatment interruptions if we had a test that could actually tell us whether drugs were working or not. So that's what we're going to be talking about on August the 13th, same time, 1 p.m. Eastern time. And what is that? In other words, 10 Pacific time. So please join us again. And I hope you all can stay cool and come and see us again next time.
Thank you.
Thank you.
Thank you.
So great seeing
Bye.
everyone.
Take care. Bye-bye.